Let's recap some of the main concepts we've covered in this particular lecture. We want to explain where the idea of colour comes from, and it's a multi-step process. The first step in the process is the illuminant. That's the object that emits light which falls onto the scene. That incoming light we refer to as illuminance, and it's got a spectrum. It's a function of the wavelength, and we refer to it by the letter E. That light falls on an object, and the object has got a reflectance, which is also a function of wavelength. So a red object reflects more red light than other sorts of light. A blue object would reflect more blue light than other kinds of light. The light that leaves the object we refer to as the luminance of the object. And the spectrum of the luminance is a function of the spectrum of the illuminance, the incoming light, and the reflectance of the object. That light enters the eye of an observer, and within the observer, at the back of the observer's eye, are a number of different light-sensitive cells. They have different responses, and they respond to different parts of the spectrum. The response of these cone cells is denoted by M, and it's a function of lambda. So the different cone cells have got different M's. The overall response of the cone cell is the integral over the wavelength of the spectrum of the incoming light, the reflectance of the object, and the response of the individual cone cell. Within the cone cell, the photons are converted via a chemical reaction into electrical impulses which travel through to visual cortex of the observer, and they result in a sensation that we have learned to associate with the word red. There are many sources of light, and we've touched on some of these. Incandescence is the light emitted by hot objects. That could be the white-hot filament of a tungsten light bulb, or it could be the sun. Other sources of light are electron stimulation, as occurs in old-fashioned cathode ray tubes, light-emitting diodes, compact fluorescent tubes and other gas discharge tubes, and lasers. Each of these has a different illuminance spectrum. Light is a form of electromagnetic radiation, and this radiation varies in terms of its wavelength. With our eyes, we're only able to see that part of the electromagnetic spectrum, but with wavelengths between 400 and 700 nanometers. A lot of light emitted by common light sources, particularly the sun and incandescent light bulbs, is in the infrared part of the spectrum. And although we can't see infrared, we can feel it, we can sense it as heat. In the retina of our eyes, we have a large number of light-sensitive cells. There are rod cells and cone cells. Now, it's the cone cells that give us our sense of colour vision. There are three different types of cone cells. So those that are sensitive to red, those that are sensitive to green, and those that are sensitive to blue. Oftentimes they are referred to as the short, medium, and long wavelength cones. The cones have somewhat overlapping response functions. In particular, the red and green cones are significantly overlapped. The fact that we have three types of cones means that we are called trichromats. We have three colour vision. Not all animals have three colour vision. Most mammals only have two colour vision. We call them dichromats. Many other animals have four colour vision. We call them tetrachromats, and birds are a typical example of that. When we create cameras, we borrow a lot of these principles. We have an underlying array of silicon photosensors, and each of those is sensitive to a wide range of colours. To make them colour sensitive, we print colour filters on top of them, and generally in a 2x2 array, which we refer to as the Bayer pattern. So each 2x2 array contains two green filters and a red filter and a blue filter. So we create in silicon an analogue of the colour sensing mechanism in the human eye. Now, if I look at an object with a particular colour and I change the intensity of the illumination, the appearance of that object will change. And here we can see examples of the object at 100%, 75%, 50% and 0% brightness. The RGB values are changing, but the colour of the object itself has not changed. So we want to find a way to disentangle colour from intensity, to map the three, what we call tristimulus values, that's the red, green, blue value, into a brightness value and two colour values. Now, we can do this very simply by converting the RGB values to what we call chromaticity coordinates, indicated by lowercase r, g, and b. Now, these three numbers are actually redundant. I can compute any one of them given the other two. So we only need to use two of them, and typically we keep only R and G. These numbers vary between 0 and 1. So if we make a graph where on the horizontal axis we have the R coordinate, on the vertical axis we have the G coordinate, then every possible color appears within this triangle. Pure red appears here, pure green appears here, pure blue appears here, and it's not possible for any colors to exist above the blue line.